DNA replication. Now, DNA replication is a semi-conservative process. And what it means is, let me just annotate quickly. So we have the genetic information, and during DNA replication, we're making copies of the existing information. Now it's semi-conservative, and what that means is these two strands, they split, and after splitting, we have the creation of two new strands. So at the end of replication, we have a new strand which is in red, and we have an old strand which is in blue. Right? So this is what semi-conservative means. Okay. Now, DNA, it requires several things in order to occur. And the first of these is a strand of DNA that will serve as a template. And as you saw in my little attempt to make an illustration just now, there are two strands that it separates, and then both of those strands, they serve as the templates. So in addition to the strand, it needs substrates, and we call these substrates deoxyribonucleoside triphosphate. Now, what is a deoxyribonucleoside triphosphate? So let me quickly try to annotate once again and show you what this is. So deoxyribose, deoxy, it's a pentose sugar. So you have the pentose sugar. Now the pentose sugar is attached to a base, all right? And so it's attached to the base. Okay. And it's also attached to some phosphates, right? So this is the deoxyribonucleoside, and there are 3 phosphates. So let me just add those phosphates. So this is phosphate 1, phosphate 2, phosphate 3. Okay, so these molecules that are floating around in the cytosol, these are what actually in the matrix of the nucleus, these are what actually serves as a substrate, and we call them deoxyribonucleoside triphosphates. Now let me stop annotating so I could go to my next so I can go to my next slide. Okay. Now, when it comes to these substrates, there are different kinds, and of course there are different kinds because, as you know, when it comes to the bases, let me just change the color once again. When it comes to the bases can be adenine, it can be cytosine, it could be guanosine, or it can be thymine. And so because it can be, and because the bases change, so does the name of the molecule. Now, if the base is adenine, then we say it's a deoxyadenosine triphosphate. So if the base here is adenine, then we say it's that. If it is thymine, then we say that it's deoxythymidine triphosphate. If it is cytosine, then we say it is deoxycytosine triphosphate. And if it's guanosine, we say it is deoxyguanosine triphosphate. phosphate. So the name varies depending on which base we have attached to this molecule here. So there is a variation in the bases and hence the name of the molecule. Now, if you look at this molecule here, we would have looked at the fact that, well, we looked at this in your first year, that when we have the, okay, let me annotate here. So when we have the base and we have it with the sugar, we say that this is a nucleoside. And when we have everything here together with a phosphate, then we say it's a nucleotide. When we add another phosphate, this molecule, we say that it's a nucleoside diphosphate because there is two phosphates here. And when there is three phosphates, we say that it is a nucleoside triphosphate, and hence the name deoxyguanosine triphosphate, for instance. Right, now I hope that clears up the name for you guys. So in addition to the template strand and to the substrates, the polymerase is also important, and the polymerase is important because it actually brings all of these substrates that I listed before, it brings them to the template strand. So without a polymerase, it's impossible to create the growing strand. And lastly, it needs a source of chemical energy to drive the synthesis reaction. So I shouldn't have deleted my illustration from earlier. Let me just make it back quickly. So remember, we have the sugar and it's attached to the phosphate. And so during the reaction, when it's basically using the substrate, remember it's breaking these bonds here. And when these bonds are being broken, we get the chemical energies here that helps to drive this reaction. Okay, now there are three things that you should know before going into the replication process. So let me just use the illustration to explain the first one. And the first one is that nucleotides are always added to the 3' end of the growing strand. Now let me just show you what is meant by that. If you look here, you can see the unwinding and exposing of the DNA strand. And the enzyme that does this is called helicase. If you look here, you can see helicase unwinding the strands. In addition to that, you can also see that we have some proteins here. These are called binding proteins, and they basically prevent the strand from rewinding. That is why they're important here. So if you look also, you can see that we have the 5' to the 3'. This is one strand, and the next strand is anti-parallel. It's 3 to 5. And so the first point was that nucleotides are always added to the 3' end of the growing strand. So if you look here, these here, let me just annotate it quickly. So these are the growing strands, right? Now the nucleotides, which we get from the triphosphates, those are the substrates, those are added at the 3' end. So that is what that point basically means. Now, the second thing is that energy is released when the bonds that links the phosphate to the deoxyribonucleoside triphosphate breaks. And so I would have mentioned this earlier when I did that little illustration there. And only two of the bonds, two of them actually are used for energy. The remaining one, it becomes a part of the sugar phosphate backbone, sorry. And that is the three things that are important to remember when you're looking at this process. And so I would have explained this before. And we can also see here we have the primase coming in. So primase is an enzyme that creates a primer. And a primer is a short segment of RNA that is complementary to the DNA. So the primase creates the primer you're seeing here. And when it is done creating the primer, then the polymerase will come, attaches itself to the primer. After it attaches itself to the primer, it will start to add the nucleotides to the primer. And as I said before, it will add nucleotides in a 3' direction. The arrows are showing the directions that nucleotides are being added. So whilst it's adding these nucleotides as well, another important thing that it does is it reads the bases, and it also replaces incorrect nucleotides. So it does a bit of correction as well. So the polymerase does numerous things. Now if you look, you can see the strand continues to extend. And it is important to note that there are names that are given to both of the strands. So we have the leading strand, and the one at the bottom is the lagging strand. So the leading strand is basically the strand synthesis continues in a 5 through 3 direction, as you can see here. And when it comes to the lagging strand, it is basically being elongated in this direction. And so if you look, it comes to the end of the DNA here. And because it comes to the end of the DNA here, what happens to the lagging strand is it starts to create fragments, and we call these fragments Okazaki fragments. And so what happened is, in order to get the genetic information from this entire strand, a lot of primers then appears throughout the strand. Now, when it appears throughout the strand, the polymerase would obviously try to synthesize the new strand by adding genetic information to the 3' point. But when it does that, as you can clearly see, it comes to a point where it will end here once again. And so it creates yet again another fragment. Another fragment, right? Okay. So you see it's there again, it's at the end once again. And so another primer is added, and then it continues to elongate, creating fragments. Now, if you look here, you can see the next thing that happens is we have the polymerase 1 coming in, and the polymerase 1 is responsible for removing the RNA primers from the strand. After it would have removed those primers, we have the ligase coming in, and the ligase actually forms the bonds between the sugar phosphate backbones, between the bases and the sugars that are in this new strand here. This here is a summary of all the enzymes that are used in the replication process, and so you can go through these in your own time. It's just some additional information. All of it is within the notes, just a little summary of the enzymes, right? So I spoke about the ligase, the helicase. Oh, and in addition to that, we have the proteins here as well. And we have once again a little description of the enzymes. So like the polymerase, it matches the correct bases, it joins the adjacent nucleotides, the primase produces the primer, the ligase, it joins adjacent strands. Then we have the helicase, which unwinds. We have the the binding proteins that keeps the DNA strand that basically keeps the DNA single stranded, so it avoids the strands from rewinding after the helicase would have melted it or separated the strands. And we also have two enzymes that I did not mention earlier, but they're important to note. The first one is the gyrase, and this one is a topoisomerase. And what it does is it relieves torsional strains that exist within the DNA molecule. And lastly, we have telomerase, and this one finishes off the ends of the strand. And that brings me to the end of this lecture.